Let's go ahead and get started. My name is Jessica Vionis, and I am the Director of Marketing at Golden Helix. Today's topic is sure to be an interesting one as we have 750 people registered. And it is my pleasure to introduce Gabe Rudy, Vice President of Product Development. Gabe joined Golden Helix in 2002 and thrives in the dynamic and fast-changing field of bioinformatics and genetic analysis. He leads a killer team of computer scientists and statisticians in building powerful products and providing world-class support. Gabe says his passion is enabling Golden Helix's customers to accelerate their research. You can follow Gabe on Twitter at GabeInformatics. And with that, I will pass it on over to him.
All right, thank you very much, Jessica. So welcome to this webinar. I'm very excited to cover this topic today as I get to kind of dive into my computer science roots. We're going to really get into the details of alignment algorithms, which is just a fundamental and must-know part of any NGS analysis. And we're really going to spend time being hands-on and cover sort of the context in which your alignment algorithm choice makes a difference. Okay, so before we get too into the beginning here, I wanted to point out that we'll try to save some time at the end for questions. I'm going to be happy to stick around and take questions for as long as you want to ask them. So feel free to start asking them as you think of them as we go through the content. That should give us a good set of questions to kick off at the end. A common question I can answer right now: yes, we will have this recorded and we'll post it usually in about a day or so. And yes, the slides will be available and we will post those as well. And yes, I do autographed t-shirts, but only when I'm forced to. No, that's a joke because Bryce kept on making me do that for some other presentation. I'll have to get him back on that one. Okay, so on the background here, I'm sure you guys are familiar with Golden Helix. If you are not, we've been around for quite a while as a bioinformatics company. We've been very proud and honored to serve the genomic research community as the technology has changed from being focused on individual SNPs with microarrays and copy numbers to now NGS. And NGS has been a focus of my development team for the last couple years for sure, and very heavily recently. We just released our latest flagship version, SNP and Variation Suite 8, which is a collection of tools for all of those types of genomic research. But in SVS 8, we really do heavily bring in a lot of power on the NGS side. We're bringing in actually the visualization engine of Genome Browse, which is another tool that we build, and we give that away for free. So you can go download Genome Browse now, but a little hint is you could also just wait to the end of the week because we're about to release a massive update to our standalone Genome Browse with Genome Browse 2. I've also been involved with a number of bioinformatics services we provided here, as well as building an RNA-seq pipeline. That gives me the unique position of having, not the unique, but sort of the vantage point of being able to have worked with a lot of the tools we'll be talking with today and kind of see how their guts come apart and go back together as you put them into automated pipelines and get them to do things they weren't designed to do and all that good stuff. So that's kind of where I'm coming from. And what I'd like to cover is a overview and then a deep dive into alignment algorithms, and then come back up and talk about sort of where they fit in the overall pipeline. What we're going to do then is start with a basic overview of alignment algorithms, and really I'm going to do this in an interactive way by showing you some of the results of an alignment algorithm, going into a visualization where we can talk about some of the details we'll be learning about today. And then we'll go, like I said, go into a kind of history of how alignment algorithms have changed with NGS analysis as its own development has changed. And as I promised in the announcement to this webinar, we will cover some of the details of how to read the output of an alignment algorithm, and finally come back for some questions. Okay, so at a high level, the analysis of next-generation sequencing is generally broken down into 3 phases. The first one just being the actual computation required to take whatever comes off of a sequence machine and put it into a standardized file format. We call those FASTQ files, and it essentially places in almost a text file a series of nucleotide letters that have been sequenced: A, G, C, T, 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 G, 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 etc. That goes into the secondary analysis phase, and this includes anything that can be done in a per-sample automated pipeline fashion to process the data. The real bulk of that is alignment, and that's what we're going to touch on. but the context of that analysis has to be put into what the overall end result is. And in tertiary analysis, this is where you bring in multiple samples, and it can be very workflow specific and can go in many different directions. Breaking down secondary analysis further, there's really going to be that alignment as a dominating factor in terms of your total compute time. I remember sitting at ASHG and watching some presentations about how I believe Baylor was putting out 10,000 exomes onto the Amazon cloud, and it was taking hundreds of thousands of CPU cycles. The vast majority of those hundreds of thousands of CPU cycles or CPU hours was just running, in their case, a BWA alignment algorithm. But we'll touch on a little bit how the line between alignment and variant calling becomes blurred, and we have to really think about what is the right appropriate roles for each of these tools, what can they do well, and what do they need to sort of delegate responsibility to another tool to do. So let's go ahead and jump into Genome Browse to take a look at this interactively here. So I'm going to go ahead and just pop open Genome Browse. As I mentioned, this is actually a production build of Genome Browse 2, which we'll be releasing on Friday. I fully expect to have our download servers broken as the floodgates of downloads begin. It's been very highly anticipated, very exciting to release it. But I'm also told that I can't spend the entire webinar going through all the awesome things that we've been putting into this. It is quite possibly the best thing since sliced bread, but I didn't get a chance to find any bread this morning, so I don't have a localized data point for that. But what I am excited to say is we have a lot of things in here. I'll point out a couple of them as we go through the webinar. And we have a couple of blog posts coming out. One actually is already out about the Evernote integration, which we'll be using to kind of demonstrate. Okay, enough about Genome Browse. What we're looking at in is alignment, right? This is a single alignment of a single read onto the reference sequence of the human reference. It happens to fall in the intron of a gene that is based on data coming out of my exome, extracted from my blood and sent to 23andMe, or actually, sorry, my saliva. So this alignment is very, very simple bioinformatically, and in fact, if all of them looked like this, we wouldn't be talking today because it would be just a done deal. In this case, it's a 75 base paired read, and you can kind of see the letters here. We're looking at a single read here and then sort of a pile up of reads there. And we can see where it fits into the human genome and what position it's at. If I click on anything in Genome Browse, you get context information of what you clicked on. In this case, we have the name of the read. Reads get unique names assigned to them by the machines that are generating them, and a really high mapping quality, which generally says there's only a 1 in a 10 to the negative 6 chance of this being incorrectly mapped to the human reference. So we're actually going to touch on, for example, what a CIGAR op means and some of these other things that look like, useful information here. There's per-base quality scores, etc. So we're going to touch on those. The other thing to keep in mind, though, is even as this looks like a relatively simple mapping, it goes straight to the human reference. The human reference is massive. If I jump out to the whole genome view, this human reference is 3.1 gigabases, billion bases long, and I'm trying to map just this individual read into this one little portion of this chromosome. So now I'm just jumped on back with my navigation buttons to get back to here. So the other thing to keep in mind is that we actually have one forward mapped read and one reversed mapped read here. What that means is you can actually have your reads on either side of the DNA template come off the sequencer, and you just have to do the template flipping to match it to the reference, which is only going to be on a single strand. So when this was mapped, it was flipped. The other thing to keep in mind is that reads generally come off in pairs on Illumina and ion torrent type sequencing, because that gives the 75 base pair read a little more oomph. The reason being, it actually came from a single template of my DNA that was about 100 or 200 base pairs long, and that template had it sequenced from one end inwards and then sort of flipped around and sequenced from the other end, and there's this little gap in between. In Genome Browse, you can use these on-plot controls to do things like show the paired end mappings between reads. So we can actually see that these two reads are paired, and alignment algorithms take that into consideration. Okay, so I have a couple reads by themselves. This is actually an exome-based data set, so as I zoom around, you can start to see I have a big old stack of reads right here covering this exon, and then right in this intron is another example I'm going to zoom in on Okay, so this is why the alignment algorithms are something we're talking about today, because these simple reads that have no changes to the reference are the easy ones. The harder ones are where there are legitimate differences to the human reference genome. In this case, the reference is a C, and there's a mismatch, a single letter change, to a G. And if you stack up a bunch of those changes, you can see on average I have 12 Gs, or I have 12 reads at this region, and 7 of them have that G. Now, just mismatches themselves aren't too hard to handle, but there are legitimate reasons why you have what we call gaps, either gaps between the sequence that was read from my DNA and the reference, or between the reference and the sequence. And if it's the first one, we have an insertion. If it's the second one, there'd be a deletion in the sequence itself. And so we can draw those and hover over those and see browse, but an algorithm has to figure out how to take hundreds of millions of these reads and place them on the human reference of 3.1 billion length string, essentially, and do that efficiently so you're not waiting weeks. And that's what we're going to cover next. Okay, so really this is all about an efficiency problem, because some of the basic algorithms have been understood for a very long time, and it's basically just a search and placement algorithm. When it comes to the alignment algorithms that do know how to handle these gaps and deletions and mismatches and these biological things, there's a number of different classes in biology. A multiple sequence alignment is any algorithm that can take 3 or more sequences and compare them. It can be sequences of nucleotides or potentially amino acids. So in this case, we're looking at comparing sequences of different amino acids at a gene level between many species, and you can kind of see which amino acids are evolutionarily conserved and which ones have gaps and deletions and differences, etc. A phylogenetic alignment is actually more concerned about what is the total, genomic distance between subspecies or full-blown species, and it understands sort of how nucleotide level or amino acid level changes occur across evolutionary time spans. Now, you can also think of a specialized class in just looking up a small sequence from your data into a very large database like NCBI's archive of sequence data. And so an algorithm's designed for that, like BLAST, are sort of its own class. But in reality, it's actually a part of this what we're going to focus on today, which is pairwise alignment algorithms. These are either local or global. A global alignment is what you might expect. It finds a total matching between both sequences. Localized alignments can allow for it to be a piecemeal-based process. Now, the approaches here we're going to touch on are historically dynamic programming and then word-based. Word-based really takes into account we've been building a lot of algorithms to process, giant gobs of text. A lot of those lexicographical algorithms, either for compression or for searching, can be very relevant when processing gobs of nucleotide text. Okay, so this is actually then how we get to some of the basis of an NGS alignments algorithms pedigree. And it starts really with this Needleman-Wunsch algorithm in 1907, which did propose a dynamic programming approach to find an optimal alignment between two sequences. Now, there's two things to keep in mind. Dynamic programming is a technique in computer science where you essentially divide the problem up into smaller problems, compute those localized answers, and often are creating a giant in-memory solution for one answer, and then you might need that to compute the second answer, etc. So it generally is uses some level of scratch space to build it up, and then once you have sort of a build-up set of smaller problems solved, you can solve the total problem and find its optimal solution. In this case, the optimal alignment between two sequences is what we call big O n times n, where n and m are the length of the sequences. What this essentially means is it doesn't scale to extremely large sequences, because you're going to take, in scratch space size, you're going to need to compute a table like this. I'm not going to get into how this is implemented. It would be a webcast in itself. But you're creating a table which is the dimensions of that table are the lengths of the two strings. And then you also need to be able to essentially make computations for each cell of that table. So it does not scale linearly. So it works. It also is very dependent on how you define optimal. In this case, if we're talking about biology, we were going to allow for things like mismatches, a single letter change between two sequences to be much more likely or prevalent or less heavily weighted than a gap. Simply because in DNA replication, we expect a single letter differences to occur much more commonly than entire letters to be inserted or to be missed in the replication. Similarly with amino acids. All right, so then in 1981, Smith-Waterman really created a localized piecemeal version of this algorithm, added a couple optimizations to the dynamic programming approach, but it's still the same class in terms of its order of complexity. So how do you go about doing something like looking up your 75 base read against all sequence data from all time, right? This is the kind of problem that NCBI tries to solve with their giant online blast suite, where you can blast the NCBI archive and look for your DNA sequence in a given species or all species or etc. Well, the approach here is that instead of doing a localized or global alignment, you essentially use a heuristical approach based on creating these substrings, what we call k-tuples or k-mers, into this large database, and then using that index to very quickly find if you have, looking at which k-mers or substrings you have in your sequence, if any of those match this large index for this database. And there's still some linear passing you have to do after you sort of localize or do a rough pass using an index. So this was, essentially proposed in 1985 and refined with BLAST in 1990. And so we've had this for a long time as well. And it certainly is something you can do with NGS data. But the class of the problem changed significantly when we started with the next generation sequencing, quote unquote, because here we are still calling it that 10 years later. But the high throughput sequencing that we started getting with like the Illumina GA2 class machine. Then these tools still work, but they weren't sufficient enough in power, or really they just took too long. Okay, so what is the class of problem we're solving with alignment? It's very different than assembly, and I am going to touch on assembly just in order to make the differentiation. Assembly is where you have no reference sequence, and you simply are taking everything that's coming out of, say, a sequencer and trying to piece all the overlapping pieces together, and then you generally put those into a graph, and you try to simplify that graph and reorder things, and you often need to use some sort of orthogonal technology to create a properly aligned full-sized genome, new, completely from scratch de novo alignment genome. This is, by definition, a very memory and time complex problem, because you potentially have to compare every read to every other read. There's obviously a lot of optimizations. I'm not going to go into that. Alignment, on the other hand, is where you already have a completed genome that you're going to work with, and that is fixed. And so you can do things on that, just like in BLAST, where you can compute an index on that and then use that index, and I don't really care how long it takes. You only create it once. The human genome doesn't change that often. And use that every time you are, taking one of these million short reads and placing it on there, and placing it in one of more places. So, like the BLAST algorithm, with the approach for NGS was to first look at, can we do something like that for placing all of these tiny short reads onto the human reference that we had. And the real breakthrough, I think, here was in the MAQ algorithm. First author was Heng Li, and you'll see his name a lot in this conversation. And I think it was a watershed not only because it sort of established a lot of the logistics of what an alignment algorithm for next generation sequencing does. It read FASTQ files, it created the idea of Phred score mapping qualities and all this kind of stuff. It also sort of set the pace and was something that people have been benchmarking against and iterating on since. Ultimately, these types of algorithms, these hash-based methods, are going to take the genome and create these tables, and we're going to simplify it here and say it's a former table, where we're just taking every unique four-letter sequence in there and then writing down the position that we see it. So let's say this GTGA occurs in position 12 and 20. Then I look in my read and I find every former that is present in there, and then to look them up in the table. And table lookups can be fast in computer science, and that's great because now we have a potential list of anchored sites that this could exist on the genome. Of course, there could be a giant list that you create here based on all the different substrings in your read, and you have to sort of cluster those and then figure out, is there likely to be a region that maps the read. But then you do the seed and expand strategy where once it's sort of, okay, I have a likely couple candidate places, and I know at least these four-letter sections are an exact match. What about the rest? Then you have to do something like a local pairwise alignment with the Swift-Waterman approach, or something even more simplified than that. But that is kind of the general strategy that has been used and extended and optimized over and over again. And in fact, even modern algorithms coming out just this year, I'll touch on these later, are doing much the same thing. They might not be using hash tables. They might be using things we'll be talking about later, but it's much of the same strategy. This overall was a huge improvement in terms of how much RAM it took to do this. With MAQ, you could take 16 gigs of RAM and do an alignment of millions of short reads, and that was a massive step forward. The next massive step forward was when the algorithms started using a technique called the Burrows-Wheeler Transform. And this was an algorithm in the 80s invented for the purpose of compression. And the reason it was in a compression algorithm is it has the property of when you transform a string, it is reversible, so you can get back to the original string. You obviously want that in a compression algorithm. You don't want to just lose your data. But also, it tends to cluster all of the repeated letters together, which allows for them to be compressed with a very simple Huffman encoding or other type of way of compression. Basically, compression always works better when things are repeated. Okay, so it happens to be that this algorithm also is a really great way to build an index into a very large string. So we're going to touch on a little bit of how it works, and we'll get to the sort of the climax of how it's used in alignment algorithms in a bit. So how it works, and this is again really fun for me to get into as a computer scientist, but I think you should. I think you will find that understanding this sort of gives an insight into how this relays into what a alignment algorithm is capable of doing and not doing. But how it works is essentially you take a string, and we're going to prefix it with the caret and suffix it with a $sign here just so we know where it is. And we can create all possible rotations. So you can think about just sort of popping one off the end and putting it back in the front, going down the row until you've done that for every position, and you have a little matrix. Then you sort that matrix or sort that set of lines lexigraphically. So you'll see that now the first row is in sorted order, A, C, G, and then all the A's have a G first, and etc. G, G, A, G, C, G, G. And we take the last column. Now this looks like a complete jumble, right? But this is what's cool about this transform, is this last column, with nothing else other than this line of jumbled text, you can get back to that original string. And I'm actually going to go through this process. And it's fundamental to the fact that BWT is a technique used in the index computed for alignment that allows very little memory to go a long way, because we can take a BWT transform and always reconstruct the original string, as well as use it as essentially this matrix is a great way to see if you have a prefix, because every possible prefix is in sorted order in this matrix. Every possible prefix, so if you're looking for a four-letter string in the data, you can binary search this matrix and jump right into the sorted position to see if it's there. Of course, you don't want to store the whole matrix into memory, and that's what the BWT transform allows us to do, because the matrix itself would be a massive squared problem. Okay, so is it possible to get from this jumbled string? We're using a simpler string here. We have no prefix character because we only need one marker for the end of the line. And this is the, again, rotated string in a matrix, pull the last column out. Can we get back from this jumbled string to the original string? A couple properties we can use to do that. First is that of the jumbled string, we know that, given that it's this column, the first letter will correspond to the last letter of the original string. So that's our starting point, is our first letter here. The second thing is that in this, we can reconstruct the first column of the matrix just by sorting the string, because that was a property of the matrix, as we sort the string. So now we have the first column and the last column. And how do we reconstruct the original string? Well, there's another property that we know that these are wraparound rotations. So this is C is always a prefix of this A, or rather this A always has a prefix of this A, of an A. That's not that useful, but this G is prefix of a C. Okay, so that's useful. And there's one final property that we can use, and this is provably correct, but you have to kind of read the entire algorithm paper to understand it, and that is that the wraparound is useful and you can navigate it for these letters here. The ith letter that is repeated of an A will correspond to the ith letter repeated here, and so then its prefix is over here. So in other words, the second C here corresponds to the second C in the first column, and then its prefix is an A. And we'll use that to reconstruct it. So here we go. We have our G to start with, and we know that this G here is the first G in this column, corresponds with the first G in this column, and its prefix is a C. So now we have a C prefix, and starting with that CG, we're here, and we're going to look for its corresponding first column letter, which is the second C. We go to this second C here, and then its prefix is an A. Follow that up, its third A, go here, prefix of an A. Okay, now it's the first A, right? So it goes up to the first A in the first column, prefix of a C. We're pushing these onto the stack here. And then this final C is the first C here, goes to its corresponding C, and its prefix is a, an A. And there we go. We have the full-blown first string. Now, as I mentioned before, we also, given that we can recreate this matrix at any time, like the fact that this is a sorted matrix of all the prefixes. And so that is essentially the way in which this is used as an index. So let's say we have an index based on a BWT, and we want to look up the string ggta. We start with the last character and find the row in which that character has a list of letters, right? So essentially, where is that in that first column? And we can represent that because it's sorted as a range. The first row to 104th row have A's. You create this index which then says, okay, for all the letters that have an A, which rows then have a TA, and you can follow down this index. So the T in the BWT sorted first column is in positions 278 to 279, and this index is computed like a tree, and so you can kind of follow down the tree, and from here you look up a G, there is no G, and so there is no GTA. So it's basically a tree or a depth first tree traversal. Now, we didn't find our GTA, so we don't give up because in biology we have mismatches and gaps. We have to decide, well, maybe there is a match to the genome. We have to account for single letter changes. Well, what we do is a backtracking algorithm. We stop at the bottom of our tree and go up a step, replace the T with a C, for example, or a T or an A or whatever, and then go down the tree again. No. Didn't find anything, there was no G. So go back up, replace the T with an A, go down, nope, didn't find anything. Okay, well, we back up one more letter, replace the first letter from an A to a G, now go down the tree with a TGG, and the rest of the string matches, and we find a match with a single mismatch. As you can imagine, this is computationally intensive, but believe it or not, it's actually so much faster than pretty much any other approach. And so the first class of algorithms that use this computed what's called the FM index, which is that tree-based structure, along with the ability to go and find where in the genome those places are that we just found. Was able to run this on a whole genome alignment in about 1.5 gigabytes of RAM, which is just immensely awesome that is possible. And that was Bowtie being able to do that. And that came out in 2009, although it was submitted for publication in late 2008. Right on the heels of that was Heng Li implementing BWA. One thing we'll touch on is that Bowtie, in its implementation, in its backtracking algorithm, did not account for gaps, so it could not do insertions and deletions. Super dang fast, able to really quickly align things, but no gaps. BWA did account for gaps, and so did a couple algorithms and many other things that followed it. This FM index is such a useful trick that even if you're not really caring so much about the backtracking algorithm, just being able to very quickly look up, a 3-mer or a 10-mer or a 15-mer is very useful. And so many algorithms that are almost more like hash-based algorithms in their approach use an FM index as their way to quickly localize sequences. So newer algorithms like BWA-SW and BWA-MEM, which we'll touch on later, do that. Okay, so that's what we're going to touch on now, is a little bit about how all these BWA algorithms are different. So BWA Backtrack, invented in 2009, right on the heels of Bowtie, was this using that FM index with the backtracking approach, accounting for mismatches as well as gaps, insertions, and deletions. It was really intended, though, for a certain length of read, and so after a certain number of differences between your read and the reference, it's going to really give up, right? It also has gone through a lot of iterations since this 2009. So don't think you're using a 4-year-old algorithm if you're not. It's gone through many different versions. Some of those versions have added a lot of features. Unfortunately, with software development, you only get the glory on the first publication, but really, on Heng Li's case, he did a really awesome job of continuing to evolve that tool into a very stable and popular, I mean, probably by default in most pipelines, tool. But a couple things to keep in mind is he was thinking about this in terms of switching out that backtracking strategy for more of a place with those hashes and then use that local pairwise matching, which gives you a better alignment of a lot of places with complex number of insertions and deletions, handles larger numbers of insertions and deletions, larger sized insertions, where you might have like a 15 base pair gap or a 30 or 50 base pair gap, which could still be picked up by the read, but you're going to need something like a local alignment to be able to pick that out, as opposed to a backtracking algorithm, which just will never stick around long enough to guess that you have to go 50 base pairs out before you start matching the reference again. All right. So then in 2013, a lot of the things he learned here, I believe, is implemented in this latest algorithm, BWA maximize exact matches. So again, it uses sort of a hash-based system and creates a giant list of places where there are at least partial exact matches and ranks those by their clustering and how good they are, and then uses a Smith-Waterman local pairwise when needed. The thing about this is it does optimize for larger read lengths, and in fact, if you have less than 70 base pairs, it's not a good choice in your reads. Most sequencers by default are at about 100, 150 base pairs coming out of the current chemistries, if not more, so it certainly is a very good default choice for that. The other thing is that it has an error rate that takes into account the sequence size. So if you get up to extremely large reads, like you might have in a PacBio machine or a molecular type chemistry, it will be perfectly fine aligning those with the fact that it might have hundreds or thousands of mismatches and gaps, as long as there's enough other anchoring matches that place it on the genome. All right, and it's also pretty darn fast. Here's from the BWA-MEM paper, a simulated set of reads, and this is the number of total mapped versus wrongly mapped, incorrectly mapped, so basically a accuracy plot. And you can see NovaAlign here, which is a commercial algorithm that has been gone through a number of, well, it's been around for a while and gone through a lot of evolution, is still the most accurate. But notice in parentheses you have the speed here. BWA-MEM is performing this alignment in about 500 seconds, NovaAlign around 4,000. So we're talking about like an 8x crazy change in speed. So really, again, there's a lot of choice in terms of speed versus accuracy here. So here's my best effort to do a breakdown of what are probably the algorithms you're most likely to run into. We already touched on the BWAs. Again, BWA-MEM and SW can handle those large reads. The other thing that they can handle is chimeric reads. So this is essentially one read that might go to multiple chromosomes or multiple entire places on the reference sequence that are megabases apart. Why would this happen? Well, potentially if you're doing, say, de novo transcriptome assembly, that might be happening. Also, if you're doing fusion gene detection for cancer, that might happen. If you're looking at structural variations, that might happen. So there's definitely a class of problems where that type of feature is extremely important. Another thing to keep in mind is that Bowtie has been followed up with by Bowtie 2 out of the group in University of Maryland. It's an entirely different algorithm in its heart. It uses a different index. It's also geared for a different type of thing. And in fact, BWA-MEM was much of a follow-up to some of the techniques in Bowtie 2 as well as previous techniques. Bowtie 2 came out first, then BWA-MEM. It's very fast though, and it's very popular amongst those doing RNA-seq. Bowtie is really probably not an appropriate choice for anything where you want to detect variants, simply because it doesn't support those gapped alignments. Also, another thing it doesn't do and this is just a matter of convenience for people who use Illumina data. Illumina data, as well as ion torrent data, tends to sort of trail off in its quality score. I wish I had a little histogram after a certain length. So even though you might theoretically read out to 200 base pairs, after 150, let's say, the quality of the base pairs drops to the point where you just really need to ignore the rest of the string. And so most tools, alignment algorithms, can handle that. And they basically, after the certain length, and they notice that the quality scores that are on those base pairs drop, they just sort of soft clip out. And I'll talk about what that means later. Bowtie does not do that. You have to be aware of that if you're going to use it. For using it for things like ChIP-seq or RNA-seq, if you're just doing quantification, you just want to know the raw number of reads that go to a given part of the genome, it might still be an appropriate choice. Nova Aligned, as I mentioned, is a commercial, run by a commercial company. It's free for academic use, licensed for commercial use. It's generally considered slow. It's got a lot of tunable parameters, so you can make it slow or fast, but the real advantage here is when you run it in all of its glory, it can be very, very accurate. Mosaic is actually an algorithm that was developed in Boston and with a lot of collaboration with the 1,000 Genomes Project, and it's going to be used very heavily going forward into the phase 3 of the 1,000 Genomes Project. They just recently published, but they're a very mature algorithm that's been developed for a long time. If you read their paper, it's a tour de force of all the techniques that we've just been talking about. They use FM indexes, they're using Smith-Waterman local alignment. They do a lot of optimizations on Smith-Waterman using modern CPUs, parallel instruction set, and all this kind of stuff. So it's actually a very impressive showing and one that's getting a lot of testing by being part of the 1,000 Genomes effort. Now, the guys who build sequencing hardware tend to write their own algorithms, and you might scratch your head as to why, but ultimately they are trying to build things for their own specific reasons. And in the case of Illumina, they have an Isaac as their latest algorithm. Before that, it was Eland. Isaac is an aligner that is much based on the hash in place and seed and extend, but they're taking advantage of the fact that we're not running this on desktops. They're trying to align whole genomes as quickly as possible, and they'll use 60, 100 gigabytes of RAM, whatever's available on the machine, and so they can be very, very quick. They tend to have lower performance for insertions and deletions. TMAP is also a very hybrid-based algorithm ion torrent developed for its own data. It does handle insertions and deletions quite well, a bit different in some of its characteristics. Generally doesn't seem to be used very heavily outside of the ion torrent suite. Okay, as promised, we're going to be able to spend a little bit of time learning how alignment algorithms describe the work it just did. Placing it onto the genome is not just about saying you're at chromosome, 1, position 100. Because of those potentials for gaps, we have to be able to describe where those gaps exist between the reference and the sequence that you're aligning. So, really, I'm going to talk about SAM-BAM because it is, for all intensive purposes, the only file format that you're going to see alignments in. Certainly not the only one that every tool ever uses, but it is the common language of alignments. And so it's going to be the output that a variant caller tool picks up. The nice thing about it is that it has two complementary representation. One, which is in text form, which is interchangeable with its binary form. The text form you can read line by line as a human if you are into that sort of thing. It's very useful for spot checking things, but more importantly, it's very, very simple to write a script to do some sort of very simple filtering or processing or transformation of it. Its specification is defined in the SAMTools package. You can go to SAMTools' homepage and find it. Written by Hang Lee. It was along with the BWA algorithm. It came out with the BWA algorithm as BWA matured into popularity as the file format is really designed and written by Hang Lee. Been having a number of implementations outside of the SAMTools kit, and of course used in almost every tool now as a standard output format, at least in its text form, and then you could always convert it to binary with SAMTools. So we're going to touch on the CIGAR string next. The other thing to keep in mind is there's a field here which is a arbitrary key value pair. It's not required, but it can put in there pieces of information that might be useful for the user, for the variant calling algorithm. There's this code, which is a two-letter code followed by the type of data followed by the data. So NM, I, and then 1. And so there are standard codes for specific things like, say, read, we'll touch on a couple of those, such as the number of matches that that read has to the human genome. So some of those codes are very useful, and you might use them for filtering. Okay, describing a CIGAR string. So I have a read here, 75 base pairs of my exome again. I've highlighted it. And one thing to keep in mind is that we have to account for these gaps in the CIGAR string, and without that we don't actually even know what the length of the read against the reference sequence is, because again the SAM file just has the chromosome start and then the full-blown set of letters that came off of the sequencing machine. So with this CIGAR string we're saying something like, okay, there's 4 letters that are in sync with the sequence, then there's 1 letter where I'm gapping or deleting against the sequence, and then I have another 72 letters where we're in sync with the sequence. We use this M, or alignment algorithms tend to use this M, because they don't care so much once they've placed it, whether it's an exact match or a mismatch to the DNA nucleotides. There are codes to represent that, sequence match and mismatch, but generally you see M used regardless of that. And then we use insertions and deletions to account for differences in lengths and gaps between the two. These other ones are relatively informative under specific contexts. N is used to skip regions in the reference, and you'll see this when aligning reads across intron gaps for RNA-seq data. Soft clipping, we covered that. That's actually when you just say, I'm not going to align the rest of this read because the quality is real low. Hard clipping is generally only going to be present when we have high quality data here at the end of the read, but it simply does not fit. We feel like it's not worth showing you that every single base is different from the reference. So we cut it off because we believe there's something else going on. Maybe this read spans a breakpoint of a copy number change or a structural variation, or we're in a chimeric region. So those reads that are chimerically aligned sometimes show up as 2 reads with different hard clippings, 1 on each side. Padding's not generally used unless using multiple sequence alignment. We already touched on those. Okay, so now I'm going to touch on how algorithms themselves can make a big difference in an outcome, and put that in the context of interpreting a variant that is actually clinically significant. So to do that, I'm going to go back into Genome Browse here. I'm going to hide this console so we have a little more space. This is my own exome. I'm going to pull up a view here of a attached Evernote window. So this is, we have embedded in Genome Browse 2 the ability to write your own notes. This is our own implementation of an Evernote editor, and it's a cloud synchronized format so that I can actually share this with friends and publish it as HTML, etc. In this case, I was using it to record interesting sites, and one of those interesting sites I placed here as a bookmark, so if I click on that I can jump to it, and I also had some images I saved in there. I'm going to close that down now so we can just look at this site. Okay, so this is the site that I found was interesting. The reason being, I'm right here on the tail end of a exon and going into an intron gap, and I have a clear 1 base deletion in what looks like probably half of the reads supporting my coverage here. So the variant caller said that I have a heterozygous deletion, and given that this is a functionally significant gene, that's interesting. Thankfully, it's not a homozygous deletion. It would have clinical implications. But I was suspect about this for a number of reasons. If you zoom on out, you can see the number of mismatches here are quite high, and I have pull in some more tracks, like say dbSNP. So although this is cataloged in dbSNP, it was not cataloged in the 1000 Genomes project. So it's like, okay, why do I have such an interesting rare variant here? Well, I pulled in data for my own exome aligned with a different alignment algorithm. This was a real-time genomics aligner. This is a commercial company that was building algorithms designed for clinical grade variant calling, and they kind of have their aligner and variant caller very tightly coupled. But the point is, the same DNA coming from my saliva sequenced with Illumina HiSeq and the same FASTQ files resulted in a very different set of alignments. Essentially, you can see that none of those mismatches that we see here are showing up in the real-time genomics alignments, and it has to do with the fact that all of the mismatches here happen to be in reads, and you can see their haplotypes pretty clearly here, that have quite a few differences to the reference. And if I click on this, let me pull up my console again, so we can click on this read. You'll see some of these properties we've been talking about, such as the adjusted CIGAR string. There's lots of single base changes, a deletion here, and the overall, there's an edit distance score. The edit distance is quite high to the reference. In other words, if you were to compare the two strings right beside each other, they'd be 26 letters different. But BWA does give this a high mapping quality, so there's no obvious filter in which to remove this read. I'm not going to go into the story of what's going on, but what it comes down to is that these reads in my DNA are not properly placed onto this region of the genome. In fact, there's no place to place them on the genome correctly, and BWA is just doing the best job it can here. It actually goes to a sort of island of DNA that's not incorporated into GRCH37 or 38, but if you go to NCBI, you can find that alternative human assemblies do have almost perfect match for these reads that are not, that have so many mismatches here. So in other words, it's a complete false positive artifact, and BWA, with its ability to handle lots of differences, this is BWA-MEM, by the way, BWA standard has the exact same behavior. I can show that. This is the BWA that we had from my actual 23, or from my 23andMe delivery, has similarly those gaps in there. So at the end of the day, this is something that has to be accounted for, and many other types of variant artifacts have to be accounted for, and I'm going to cover those in a little bit. One could argue that one just needs to change the parameters for BWA-MEM to not align these reads which have so many mismatches. I went back and tried to do that and utterly failed. I changed a couple very obvious parameters, filtering thresholds, so probability scores. Again, the probability, the alignment scores here are quite high, and so there's this issue with alignment scores being very difficult to compute in a way that's meaningful. And in fact, modern algorithms like Mosaic actually have a whole AI-based backend purely for calibrating empirically and computing these alignment mapping scores. They have a neural network-based system for that. So that's a bit tricky. And so getting into a discussion of, okay, what are the, where are the places where we still have problems with variants themselves? And I'm going to break it down into these 4 categories. So we have problems where there are issues with the reference assembly. That could be that the sequence that I'm looking for is underrepresented. Essentially what I just looked at, there was a sequence that was not in the human reference, and by the way, it's not in GRCH38. It just simply has not been placed into any of the human assemblies as primary or alternate sequences. Or you could have tiling issues where these back clones that they use to construct the finished genome have overlaps in places with, essentially one haplotype switching to another, etc. But more importantly, with the human reference genome, we have to keep in mind that there are so many repetitive regions. In fact, given some definition of repetitive, over half the genome is repetitive. And there's going to be lots of regions with very low, what we call sequence complexity or information density. This ultimately results in, no matter how good of an algorithm you have, you cannot uniquely place your read onto the genome. And if it's not uniquely placed, we generally throw it away for variant calling, because if it can go to 2 places, you don't want to necessarily trust that for informing a very localized mutation. So this mappability is a big issue, and regions that sort of kind of go up and down in mappability might have a 60 mer repeat, and you have a 70 mer string. You might have the tail end of it coming in and doing quite well, and then the rest of it getting messed up and that kind of stuff. Also, structural variations in your own sample data can confound alignment algorithms that are based on very small reads and looking for very small gaps, right? So imagine you have a giant deletion that's millions or megabases large. Within that, or at the breakpoints of that, you're going to have very confounding alignments for things that don't properly match into those deletions. Similarly, mobile element insertions, inversions, translocations, these will all cause problems. So none of these, in my mind, are something that we can blame the alignment algorithm for. They're very complex issues with the biology that we're dealing with, and we generally are okay with dealing with these because, thankfully, the most informative functional portions of the genome, at least in the bulk, easy-to-access sense, are not in the extremely highly repetitive places. The next thing is, complex variants. And what I mean by that is any locus where you're having potentially multiple insertions or deletions or SNPs all within, say, 10 or 30 or 40 base pairs. And so the way in which you describe that can vary from one alignment of a single read to another alignment of a single read, from one algorithm that calls variants to another. So this is kind of gets into where is the responsibility for, say, tackling that last problem, handling complex variants. The alignment algorithm, we expect to place reads into the right part of the genome. Okay, check mark. We also expect it to give high quality mapping scores, very useful for being able to, say, potentially filter out noise into a variant calling data set. But when it comes to variant calling, there's all these other things that we potentially need. We need to be able to, look at whether the mate pairs that are around it make sense for structural variation. We need to know the multi-mapping information. And we need to essentially look at multiple alignments at a single time. Remember, an alignment algorithm takes one read, uses a giant index, places it on a genome, forgets about it, goes to the next read. A variant caller looks at a given region of the genome, looks at everything that's out there, and so it really ends up being its responsibility to account for all of these differences that we can see and ways that data is simply represented. Okay, so I touched on GRCH38 a little bit. Long story short, I don't see this as fixing a lot of these issues. At least it is fixing issues, and I have to say that it's a lot of effort and a great thing that we haven't. We'll be moving to it. It's just going to be a slow and steady process. I aligned my own exome against 37 and 38. You can see the number of SNPs goes down, the TSTV goes up. So there's certainly a class of false positives, in this case about 10,000 or so, that are being reduced or removed by this change. But that last class of problems is really not going to be fixed by the human reference. So what do I mean by multiple representations of the same alignment? This is a perfect example. So here we have two samples, but they actually have the same haplotype at that region. So if I'm going to read this haplotype left to right, it's GG. I'm going to just read the haplotype that has this this mismatch in this deletion. Right? So it's a GG, TT, T, G, TA. If I read it on this read, it's a GG, TTT, G, TA, right? So it's the exact same thing, and you can describe it because there's this 4-base homopolymer of a T in two different ways. And this is the same alignment algorithm, just happened to place it in one place versus another. This could be based on a number of multiple reasons, but is it the alignment algorithm's fault, or really you have to have another tool who takes this all into account and aligns them? This was actually already locally realigned. So in fact, all of the reads, there's about 25 of them, have a consensus on them being placed in this position versus this position, but ultimately it's still the same haplotype. So it's bigger than just getting them, to agree between individual reads. You have to start to look at a larger class of problem. And how you describe variants is another one. So, we have a consistent placement of a TCT deletion at this locus, but do you describe this as a TCT deleted at this position, or TCT deleted at this position? So there are multiple representations that are all legitimate. Thankfully, we have a general policy that's been de facto standard in variant calling of always placing things in the leftmost position of simple repeats. Simple repeats is one thing, but as our previous example shows, technically this is correct. This is the leftmost position for this deletion, and this is the leftmost position for this C, but it just happens that they are different representations of that haplotype. Similarly, when you talk about complex variants, you can treat them as a larger string replaced with another string, or individual mutations. So in this case, this is another place on my my genome. I have a G or C GGGT replaced with an A GC right? So some of those are still reference positions, but some of those are mismatches. As a haplotype, it's useful to treat this as one giant mutation because you can kind of look at it in terms of these mutations are all on the same chromosome, hence they functionally are going to behave only on one chromosome. You're not having SNPs on both chromosomes. As individual SNPs, if you don't have phasing information, you don't know whether this is on one chromosome and this is on another. Yet at the same time, for annotation and for really utilizing any level of comparison between one file and another, we generally place data into its most primitive allelic form, such as this. And it would be nice if they could be able to go back and forth. And if you have a good phasing information stored with these mutations, you potentially could. The tool used to do this, by the way, there's a number, but the most popular, I would imagine, is called VCFlib, and it has a command called allelic primitives, which will take this VCF file and essentially create this VCF file that we're looking at in Genome Browse. So the guys that I really respect the most in this field for doing this kind of work and shining the light onto this class of problem and really figuring out which are the problems that are tractable and how can we as a community start to agree on things like data representation are probably the Genome in the Bottle folks. So this is a NIST-sponsored organization, and Justin Zook's probably their lead bioinformatician. Is the first author on their Nature Genetics paper that they just recently put out. I really would recommend it for anyone who wants to dive deeper into this. What they're doing is taking a single sample, a single cell line, and sequencing it on every imaginable platform with every imaginable sample prep and every imaginable variant caller and alignment algorithm, and then creating a consensus variant set with as many heuristics and good tools as possible to give us essentially a really high quality true variant set, as well as being able to create these masks for things like regions of the genome where calls end up being very discordant. Being discordant because we're in a class of problem that we haven't solved consistently across all the tools. So those are all very useful. This other resource that they even refer to in their Nature paper is ggta, which allows you, if you have your own pipeline, to download a set of that same sample, various different formats, run it through your pipeline, upload the results, and then compare your results versus other algorithms. Let me show you what this looks like a little interactively here. I'm actually an advisor on this, by the way, if you go to advisors. So I've played with this a bit, or at least had the chance to talk to the guys who built it. It's a great platform. So in this case, I have 3 different alignment and variant callers stacked up against each other, and you can see sort of their Venn overlap, their true and false positive rate based on that Genome in the Bottle truth set. Here you have a ROC curve. You can see that BWA-MEM with the GATK unified genotyper does quite well. Nova Online with FreeBayes a little bit worse, but still quite well. BW2 not so good on this exact data set. Okay, one final thing, and then we're going to get into questions, and that is to show you some of this Genome in the Bottle data. And to do that, I'm going to switch projects here. Go to my recent Genome in the Bottle examples. This project is loaded entirely from yet another new feature in Genome Browse 2, the ability to add sources from URLs, and I created a little HTML file with all of the S3 hosted Genome in the Bottle samples. These are massive samples. If you look at them size-wise, in the order of 200-300 gigabytes each, and we're loading all of these into this one Genome Browse view, and I'm using that Evernote integration to be able to take little screenshots. What I'm essentially doing here is going through the figures that they put in the supplementary section of their paper and getting to the same places and seeing if I can kind of recreate those figures and explore them on my own. So I can kind of zoom around here and look at that. So this is, for example, a very complex variant that TMAP and BWA have a very different way of describing. TMAP places all of the insertion up front and then a couple mismatches. BWA has a couple mismatches up front, and then there's this giant insertion in the tail end. Again, this is the exact same haplotype represented multiple ways. Let's see, there's another example here in my notes I want to go through. Here's another example of a class of variant that can just be confounding to align and call. In this case, it's a very clean set of variants. I could split this by strand. It's a little more clean to see forward and reverse. The C and the deletion are each on different chromosomes, and yet the TMAP algorithm has a very hard time picking up that deletion. It's putting something over here, but it's not even that deletion, and doesn't end up calling that SNP. So you can see it's very well covered across forward and reverse strand. And finally, here's another example they have in the beginning of their paper. So I'm clicking on these little bookmarks. Another thing in Genome Browse 2 is you can take a given zoom window and say, this is a region I like, and then create a little bookmark for yourself to click on later. And this is a complex variant. Again, it's a TAG TCA replaced with a TC TCT. And you can see that the aligners have a very different agreement. Let's go down. So here's some very nice, high-quality 200 base pair paired-end reads aligned with BWA. Here's some ion-proton, 200 base pair data as well. It places the insertion way downstream, right? And it's not even a homopolymer, so I'm not quite sure how it figured that out. And here is a solid 4. Solid has a hard time handling this region at all. The coverage drops dramatically in this region. And here's Nova Align. I'm not quite sure what Nova Align is trying to do here. It looks like a giant mess to me. It doesn't seem to pick up the insertion. It kind of just shows a bunch of mismatches. So it's having a hard time with that 6 base pair insertion. So in this case, the winner goes to BWA. Certainly not everyone does, but if I were going to recommend an algorithm as a general first pass on any variant calling stuff, it would be BWA-MEM with GATK's new haplotype caller. Seems to do the best in terms of just how this genome in the bottle is reporting a lot of their variants. Okay, I clearly went to the end of the hour because I'm so excited about showing you all this stuff. So sorry I didn't leave more time for questions, but please stick around because I'm ready to stick around myself, and we can answer as many questions as you want. Otherwise, thank you for joining us today, and Jessica will take it from there.
Thank you so much, Gabe, for all of your hard work really researching and preparing this topic for us today. I found it very interesting, very geeky in terms of the alignment algorithms and how they're actually calculated. If you didn't have a chance to type in your question, go ahead and do that now. We'll stay on, maybe for another 10 minutes and answer some of these questions. As Gabe mentioned, version 2 of Genome Browse will be available on Friday on our website. But if you want to receive an email about that, please respond to the survey after this webinar. It just pops up when you close the window. Okay, let's go over to questions.
I'm going to flag one here.
Ooh.
So do they really start with formers is the question. Of course not. I was actually very simplifying the example with a former. You would never actually index the entire genome with formers. I'm not quite sure what the default key size is. On a modern algorithm, I'm pretty sure it's close to 30, sometimes 15, but more like 30 for indexing. And obviously I was creating a simple example for us to walk through there. Okay,
thank you so much, Gabe.
Yeah.
Hey, you want to tell us a little bit about Top Hat and Top Hat 2?
Sure, absolutely. So Top Hat as an algorithm is really sort of a meta compiler. It was using BWA in its first iteration to do the back-end work, and on top of it, it's doing things that are sort of aware of how RNA-seq gets spliced across in genomic space and RNA space. Top Hat 2 uses Bowtie 2 to do the same thing, I believe, and really it's actually, I believe it actually handles both Bowtie and Bowtie 2, but it's just an evolution on the same concept. So this is an entirely different, topic I could cover, and because I would say the default algorithms of choice would be very different on an RNA-seq example, especially if you're trying to detect novel splice sites. That is, by definition, a problem that's very tricky for short reads to do. You have to do some of that chimeric read type stuff where you don't know that there's a 3,000 base gap between the first and the part and the tail end of your read, and you have to account for that. And so there's a lot of techniques to account for that. Different aligners like STAR are actually very popular for RNA-seq, as well as, I'm trying to remember them, GMAP, etc. So there is sort of a different set of considerations when handling RNA-seq data.
You mentioned GMAP. Is that the same as GSNAP mapper?
Yeah, that's what I meant, GSNAP.
Okay.
Right. Yeah.
Because there was a question, do you have any feedback on it?
I haven't had a chance to use it very much. I have had plenty of people talk my ear off about it, so it's obviously got a very passionate user base, and that's the sign of any strong and healthy community and tool. So there's, what I have seen though is it's used very heavily on the RNA-seq side, potentially the ChIP-seq and stuff as well. Haven't seen it a lot on the DNA variant discovery side.
Okay.
So go ahead.
I was going to go on to the next question.
Yeah.
What if there are already SNV mutations in the seed sequence?
Right. So the seed being, well, if the seed, by the seed you mean like the reference genome, then, well, you're absolutely right. There are places in the reference genome that have a copy number, repetition. And there's places in GRCH38 that have multiple alternate locations, given that there is such a high level of variability within the human population of these very large mutations or very large haplotype changes. If you mean inside the actual short reads themselves, then you get into this issues of like, how do you map a read that might be spanning a gap or sort of a breakpoint of a structural variation, a translocation, even an inversion, all that kind of stuff. Those generally are going to be seen by the aligner. If you have enough of it anchored into the genome, they'll be placed and they'll be hard clipped right on the tail end of where essentially the rest of that read no longer matches the reference genome at all.
Okay, sounds good.
Yep.
Okay, next question. Could you improve your BWA-MEM alignment by doing a GATK realignment?
Right. So there's actually a couple questions. Oh my goodness, there's a lot of questions about that specifically. And again, I could almost spend a whole webcast covering realignment tools. One thing I wanted to point out, so in the standard sort of BWA1 or BWA backtrack GATK pipeline that 1,000 Genomes used and a lot of these web-based pipeline tools use, you're using a number of passes to essentially create the perfect BAM file that then goes into the final variant call set. And one of those is to use GATK InDel realignment. That's doing a couple things. It is making it consistent across all the reads in a site, how you describe a variant, but it also generally left shifts all of the insertions and deletions. And it can essentially create some very simple haplotypes, etc. Especially with the first version, that was about the most it can do. And it showed its work because it created a new BAM file which had all that cleaned up. And most of the BAM files we looked at today had already got that sort of pass done on it. Things are a little bit different in the modern breed of GATK's algorithm toolset. In their haplotype caller, they found it so fundamentally necessary to include a very tightly coupled interaction between that InDel realignment tool and the haplotype calling that they essentially do it all the time, and they don't show their work per se because they're looking at everything. They're constructing all these potential putative haplotypes. They're taking the fact that it's a diploid chromosome and there's two potential haplotypes, and then they sort of create the VCF file that encodes all that information, but the BAM file is left untouched and could still be a giant mess of lots of different places in which gaps are described, etc. So I think we're losing something there, and there are going to be ways in which you can kind of force it to re-encode BAM files. From what I can tell, they don't work all that well, and they're not sort of a default. But on the plus side, the haplotype caller has a much better, almost what it does is a de novo assembly at that site of the putative haplotypes and then scores them based on which ones have the most evidence and can be matched with a pairwise local alignment back to the human reference genome. So it is definitely a vital and critical thing to discuss, and I don't have more time, but I hope that kind of gave you a quick overview. You certainly need to have a tool that can do a local realignment. Another tool that does it in place during the variant calling process is Freebase, which is also used pretty heavily by the 1,000 Genomes project. It will do a local realignment on the order of doing a local assembly and then placing it on the reference. SAM Tools does a very simple local realignment of just simply normalizing how those gaps and deletions are described when it calls variants. It is MPileup. It doesn't do anything as complex as Freebase and GATK.
Okay, and then I actually had, I think, 2 or 3 people who are also asking about the star aligner.
Yes, I didn't. So again, I don't get as much time as I'd like to just sit around and run a bunch of tools. There wasn't a star alignment in the Genome in the Bottle dataset, at least not in the publicly available FTP versions. And unfortunately, I don't even see a lot of representation of star in GCAT. But you can certainly run your own comparison and pull up an alignment algorithm comparison or variant analysis comparison in GACT to benchmark it. It is probably the most popular one out there that I can tell in more of the RNA-seq and ChIP-seq side, and it certainly seems to be adequate for doing variant discovery, but I can't speak to sort of its pros and cons.
Okay. Can you talk just a little bit not a full hour, about how to align polymorphic regions such as HLA.
Well, that's a good point. So I would classify that in my sort of regions of problems, not in the responsibility of the alignment and variant calling to solve. It is fundamentally a very large-scale problem of having entire genes, having dozens of pseudogenes that are very highly similar, and a 75 base pair read is, or even a 250 base pair read, is just not going to cut it. It's not going to. You will be able to align it to all the pseudogenes into one spot or multiple spots, etc. Ultimately, with those regions of the genome that are that polymorphic, you either need a completely different sequencing technology or a different technology like long-range PCR to properly assay them. The closest thing I've seen, and I touched on this in my own exome analysis webcast, where I did an analysis of my wife's data because I was interested in her HLA region, given she has a phenotype related to the immune system, was that you can use a specialized process to essentially align against all the known HLA alleles and then rank those based on which ones have the best alignment statistics and coverage. And that's actually what Omixon does with their HLA target alignment, and they'll take exome data and run it through a completely different algorithm. You won't be able to look at it right there in your BAM pile up with all your other data because it's essentially aligning against the 300 known HLA types or whatever, and then you'll just get a spreadsheet with which HLA genotype or haplotype or which HLA type you're likely to have. So that's really the appropriate way to go about it. And if you look at HLA, I'll go back to my exomes real quick. It's a complete mess, and it's not something that we can blame on the variant caller. So HLA DR beta 1, right? There's just no way that you're going to pull across the 10 different pseudogenes that are accounting for the variable height in this histogram of read depth and mismatches and all that. So there's really a totally different class of problem. So a little more time than I need to spend there. Yeah. Let's see.
You mentioned the GRCH38 assembly. Mm-hmm. When do you think is a good time to use it as a reference for alignment and variant detection, like workflows? And what's the benefit going to be using that over GRCH37?
Right. So right now it's all a matter of what you need to do with the data, and for most people's uses, I can't imagine the benefits of going to GRCH38 now outweighing the cost of you being in a complete desert island of annotation and tool support. All of our current data is aligned to GRCH37, right? Not 38. We do support GRCH38 in Genome Browse. I think I curated it last, and when I aligned my own data to it. There it is right there. And just the number of annotations, though, are going to be essentially nil. We're going to have a reference sequence, and it wasn't even just till recently till we even had genes that were aligned to GRCH38. Compared to our current annotation repository for 37, where you'll see we have every version of dbSNP, all these functional predictions that you're going to use for classifying which variants are of highly relevance, gene annotations, clinical annotations, ClinVar, all these things, they're going to, and they really don't seem like they're in all that much of a hurry. So at the moment, there's no good reason to go there unless you're just exploring it, or you are essentially dealing with variations that are fundamentally structural, and you are fine being sort of on a desert island where you're making your own waves.
Okay, you know what? Let's ask one last question here. We've kind of gone over time.
Oh, okay.
You mentioned BWA-MEM as being good as a first pass. Would you add another aligner for clinical uses? The discordance shown by the genome in a bottle data is concerning. Is this issue partly mitigated by the fact that most disease-causing mutations are in more unique sequence segments?
Yes. I can start from there. It's a long question.
Okay,
I can see.
It is a long question.
Yes, obviously I just showed you some worst case scenarios, so don't be too scared. 99-point-something% of variations are going to look pretty dead simple, and even insertions and deletions that are not flanked by a bunch of other mutations are starting to be very accurate with the latest class of mutations. And they're certainly clinical grade. Nothing is perfect. Even existing clinical assays are going to be confounded by complex mutations and complex things in biology. So yes, I believe BWA-MEM is a good first pass. There is other algorithms to look at, and I wouldn't say you should stop there. It's certainly on the burden of someone developing a clinical pipeline to do an empirical analysis on the type of data, on the type of sequencing platform and chemistry they're using to validate that they're getting accurate in terms of sensitivity and specificity on that. Another thing NIST is doing, by the way, with the genome in the bottle, this is sort of just one front of it, is they're trying to help define these types of things in terms of, giving you a truth set to benchmark against, giving you the actual samples. You can go buy NA12878 and run your own, gene panel against that and test against the truth set. So these are all sort of all within the same camp of trying to make this data ready for putting into a clinical pipeline. And it's not about every single situation being perfect. It's just knowing what the strengths and weaknesses are.
So then, regardless of the issue with the false negatives, especially in the case of clinical false positives, what do you think about addressing it with Sanger confirmation?
Yeah, so obviously that is still the de facto standard, given that there are very few FDA-approved medical devices that are only NGS. In fact, I think the MiSeq with CFTR is the only one. Maybe that's a little out of date. But so confirming it with Sanger is still kind of the best practice for going into a clinical report. Although with certain classes of, again, so if you're targeting 2 genes or even 30 genes or 100 genes, and the sort of the confounding issues in terms of the repeat regions around those genes. CFTR's got a nasty repeat region right beside one of its exons, right? And you can handle those, and you know how to handle either masking those or accounting for the fact that you're going to have false negatives there. That's all well and good, and I think there's no reason why NGS data won't be becoming the primary diagnostic tool for clinical uses. It's just a matter of doing the proper test development.
Okay. Are there anything else you want to throw in there?
Oh, I'd like to answer more questions, but let's wrap it up. I'm very excited to get Genome Browse 2 out, and we'll be excited to hear your feedback as you try downloading it and using it for your own annotation and variant analysis and algorithm discovery process. So please give us feedback, and we'd like to do more of these webcasts, so that will help guide our decision.
And if we didn't get to your question, we'll try and make sure we email you personally.
Okay.
All right. Thank you, everyone. Thank you, Gabe.